Good afternoon to everyone. Let me do a little 360 here. We're going to change tune a little bit to an area that I've been involved in now for just over a decade. So my name is Henry Rodriguez. I happen to be the founding director of the National Cancer Institute's Office of Cancer Clinical Proteomics Research. And it's an office that's located in the director's office within the Division of Cancer Treatment and Diagnosis. And the main focus of this office when we created it is on trying to apply multi-omic based approaches, bringing those assays to clinical trials with the hope, which is what we're all trying to get, with the hope of being able to translate any new knowledge that we identify and move it towards patient care. And what I'm going to do today is talk about one of the flagship programs that we run out of the office that's known as CPTAC. It's a program that actually got created about 15 years ago, and it really was a metrology-based program trying to measure proteins on large scales. But over time, what we did 5 years ago, seeing where the genomic space was going, we decided to take every patient's tumor and now comprehensively characterize that sample always at the DNA, RNA, and at the protein level. So with that as a backdrop, let me start here my talk. Oh, perfect. Okay. So many of us, quite frankly, which is what I have to be. I mean, I actually did all my trainings, my fellowships come out of Los Angeles, and I have to be a DNA jock, but ultimately I transferred, and here's why. If I go to individual and I say, define for me what you think is precision oncology, I would not be surprised if everybody says, oh, precision oncology, that's a discipline of genomics. And you know what? I get it, especially in oncology. In oncology, today we recognize that cancer is definitely, ultimately, it's a disease of the genome. And this is going to be evident now through the major strides that have been made using genomics as a way to understand and treat numerous diseases that's associated with cancer. And actually, one of the nice ones is you can actually begin to look at real good data. Now, this is adult-based data, but nevertheless, it's really good data. You can go back several decades and begin to understand what investments that we've made through public and through private investments when it comes to precision oncology and the impact that it's starting to have. And one of those data points are nice papers that came out just a couple of years ago. And one is by looking at cancer mortality rates in the United States. So what do I mean by that? So if you look at the left-hand side of this slide, turns out prior to 1991, the overall cancer death rate in the US was always steadily rising. But what happened after 1991 is that the overall rate started to fall. And on average, it's been falling about 1.5% per year. And I should note that from 1991 to today, that actually translates to approximately 3 million lives that in theory now have been saved. But the really big news, when you start parsing through this data set, the really big news is that it turns out it didn't fall 1.5, it fell 2.2%. And that actually occurred from 2016 to 2017. And it's held steady, the last data set that we just got a couple of months ago from 2017 to 2018. And that represents the largest single one-year drop since 1990. And while this progress is going to be mainly driven by declines in death rates for the 4 leading cancers in the US, you can look at the right-hand side of that slide, which I've graphed them out there, and that's going to be for lung, colorectal, breast, and prostate cancer. What was really interesting is that declines, though, dramatically accelerated for lung cancer, which in the US happens to account for about 25% of all cancer mortality that's affiliated with cancer itself. So a lot of us at the NCI and within the academic, we started to ask ourselves, so what's going on within this data? Fortunately, colleagues at the NCI with others, they actually published on that data. And what transpired here is that we recognize that tobacco control has played a huge role in declining deaths when it comes to lung mortality of cancer since the early 1990s. But the real exciting news by looking at the data is that it turns out advances now in targeted therapies is actually what's starting to contribute to the sudden rate drop in cancer mortality, specifically in non-small cell lung cancer, which in about 2013, which are those 2 little arrows now that I've drawn on the left-hand side of that slide, such as Tarceva and Iressa, actually came on the market and they were being administered to patients, which happens to be EGFR tyrosine kinase inhibitors. And I should also note that those data points in this manuscript that was published in 2020 doesn't even begin to take into account immune checkpoint inhibitors. We're targeting PD-1, PD-L1. As a result, the good news, if you're into precision oncology space, we can even begin to expect further declines and death rates, in this case from non-small cell lung cancer in the outgoing years. So that's really good news. Yet with all that great fanfare, I'm going to make a very bold statement. And that is, I still believe that there is a disconnect in our ability to be able to connect the underlying biology of the vast majority of cancers that are out there and translating that towards patient care. I still believe that there's an inability in our capability to truly understand when people talk about the genotype and trying to connect that, which what you want to get is at the phenotype level. And I'm going to make this argument very simplistically today, just in one slide, but I'm going to make it from the perspective of cancer biology, from a very genomic-centric way of viewing it, and its translation towards patient care, which, quite frankly, it is the leading approach that we're using in structuring a lot of our clinical trials. So if I were to say cancer biology at large scale, a lot of people are going to think of one of the sister programs out of my institute, which happens to be the Cancer Genome Atlas. To be very clear, this is an incredible program, what they did from a brute force perspective. In a span of 10 years, they basically comprehensively characterized at the DNA and the RNA level tumors from over 20,000 individual patients. In the process, they actually cataloged that for just over 30 distinct tumor types. Now in the process, they also identified a lot of interesting biology, a lot of observations were made, but in those observations, they also identified actionable mutations, of which follow-on studies were performed, of which those follow-on studies, some were positive, they developed small molecules, you had targeted therapies, and they're actually running a lot of these clinical trials. That's really good, exciting news. But again, now we have data, we have time on our side. We can begin to go back almost a decade and also be looking at what's coming on in a lot of the studies that we're currently doing, early stage trials. And here's the other side of the coin now. So it turns out a lot of those tumors, a lot of those patients, we identified these actionable mutations. Some of them actually don't respond to the targeted therapy. And if they do respond, you find out in many instances, it's actually going to be temporary lived. They either develop a toxicity effect, you need to take them off that arm, or they're going to relapse to the disease itself. Why? We don't have an answer, but that's okay as scientists. But what we do know with absolute certainty is that there's a tremendous amount of still biology that we don't understand underlying these diseases and being able to connect it back up to the ladder towards the phenotype, which is what we're all trying to do. And again, I came from a DNA way of thinking. So what happened about a couple of years ago at the Cancer Institute, myself and a couple of colleagues, we started to ask ourselves, so how can we begin to make now this field of precision oncology, or as I used to tell the board, how can we make it now more precise? Now, while we recognize that precision oncology absolutely would not exist if it weren't for the Human Genome Project and large-scale cataloging activities such as the Cancer Genome Atlas, there is more and more this acknowledgement that what's starting to happen is that genomic characterization alone is not always going to provide sufficient insights into numerous molecular classifications for a significant amount of cancers that are out there. So the reality is that we started to view this space now as technologies have advanced and their analytical capabilities have also now improved, especially to go after needle biopsy cores, is that proteomics and genomics should not be seen as competitors. We started to see them more as complementary or as partners. And at the Cancer Institute, we now see this as one that holds great promise. And we see this multi-omic-based approach, one that a lot of people now might call it also proteogenomics, which is combine those two disciplines, as a way of revealing potentially not only new molecular patterns that underlines a lot of these diseases, but hopefully, which is what we want, is that potential. Hopefully, we're able to inform new approaches, not just to cancer diagnostics, but potentially therapeutics. And at the Cancer Institute, the program that leads this activity for every sample we obtain from a patient, comprehensively characterizing it, DNA, RNA, and at the protein level, is affectionately known as CPTAC. So this is sort of a cartoon structure of the CPTAC program. And today, basically, the program exists. It actually has two complementary programs. There's one that we refer to as a characterization program. Think of it as TCGA 10 years ago. You're cataloging. There's a second arm to this program, and it's a translational arm. It now is where TCGA has moved into. We partner with clinical trials. So in the characterization program here, we fund, it's 100% of an extramurally driven program. We fund characterization sites. We also fund data analysis centers. Their job, these centers, they are supposed to be working in a collaborative manner, not only to generate, to integrate, but ultimately analyze deep, comprehensive proteogenomic data that ultimately gets produced. And these are typically obtained from prospectively collected cases, where every case we will get a treatment-naive tumor along with its normal adjacent tissue. And these have been optimized, these materials, such that you're able to perform comprehensive genomic and proteomic-based measurements. So that what you're looking at is not going to be representative of an artificial workflow. It's going to be representative of that underlying biology. And in the translational arm, not surprisingly, we fund translational research centers, and they, along with those data analysis sites, again, they got to work together. But in this one, they got to be performing clinically relevant research projects in a preclinical phase, and ultimately it gets brought back to our boards, where they then are granted access to clinical trial samples if it's deemed worthy. And I should mention, one of the things that's quite nice about the program when we actually created it is that all of these activities, we have partnerships. It's all done in coordination with colleagues at the Food and Drug Administration and the American Association for Clinical Chemistry. Clinical labs are running a lot of these targeted-based assays. So this idea of merging these two disciplines really got created 5 years ago. There's a lot of boards like to say, so what have you done in the past 5 years? So here you go. I'm not going to go through all the details, but on the left-hand side happens to be the characterization arm. We were held responsible to characterize 5 tumors in 5 years. You could see we outsurpassed that. Ultimately, the consortium members were able to characterize a total of 11 distinct tumor types, where they produced 12 flagship studies in that 5-year duration of a window. In the translational arm, here it was a pilot for the NCI. We focused on 3 cancer types. What's quite nice is that they all produced very interesting findings within their preclinical phase. Already now the AML and the breast cancer studies were approved for access to clinical trial samples. Those studies are currently ongoing. And the ovarian cancer study team is in the process now of resubmitting their application to the Cancer Institute. Now, this is a pediatric conference. So before I move on to the next slide, let's take a quick look at one of those characterization studies that actually focused on pediatric brain cancer, which is a really nice collaboration between the CPTAC Consortium, also the Children's Brain Tumor Network, and the Kids First program. So this has actually been published, but a snippet is the following. In pediatric brain cancer, which is really why this study is quite nice, so it's been known at a genomic level, pediatric craniopharyngioma patients happen to harbor beta-catenin gene mutations, while adults, they happen to have BRAF mutations, a very specific one. The result of that on a clinical level is that BRAF inhibitors are not treatment options typically for these pediatric patients. But what made this study, this comprehensive study, quite unique, which started to blend now genomics and proteomics at the same time, is that the study actually showed, and they demonstrated, they revealed now by looking at over 200 tumors, they span 7 histological types of childhood brain cancer. They actually identified a very novel subgroup of pediatric craniopharyngioma patients that happens to have or share proteomic features with pediatric low-grade glioma that happens to have a BRAF mutation, despite those pediatric craniopharyngioma patients actually being wild type at the genomic level for BRAF. So the implication of that, which was quite nice, is that it seems to be a rewiring at the functional level versus the DNA level. And the result of that is now based on the protein expression, that the signaling networks in these pediatric craniopharyngioma and in tumors that they happen to have BRAF mutations actually appear to be related, and potentially now could be treated with targeted therapies, such as MEK inhibitors, which have been used successfully in low-grade glioma pediatric patients harboring those BRAF mutations. And what's quite nice is that the investigators of this study has taken the preliminary data to now be in the process of actually moving that into a very early stage clinical trial. And again, to be very clear, these samples underwent DNA, RNA, and protein-based characterization. So the obvious question is, so what did the RNA clustering deliver? Well, I could tell you the RNA-based clustering, unfortunately, did not capture, in this instance, those types of novel subtypes. So, because it's a very short talk, I think one of the things that's quite nice about the CPTAC program, in fact, its mandate is just like these foundations, all the information that we produce, we give it back in the public domain. So here's a nice slide. It kind of demonstrates we give away all of the molecular characterization data. We develop targeted-based assays. All the SOPs are given away to the public. Even physical reagents that increases the sensitivity of our assays, monoclonal antibodies, we give it all to the public. And as you can see, I'm not going to go through the stats, but the point here is that these materials, these resources are used both on a domestic, especially on an international level. And lastly, if there's any hesitation to the Cancer Institute's commitment to this emerging field or exciting field for me, which begins to combine these two disciplines of precision oncology. So colleagues and myself, we recently published a perspective article in the journal Cell. And here we're examining now cancer through the lens of molecular characterization classification. Quite frankly, we're basically making the case that this idea of this multi-omic proteogenomics needs to finally be fully integrated, not just in cancer research, but in clinical trials. I started my presentation with this slide that cancer is a disease of the genome, and I actually do believe it is. But I hope I've been able to sway a couple of individuals that while it is a disease of the genome, to better understand its many diseases and quite frankly, to accelerate the development of new diagnostics and therapeutics, we should begin to study cancer as a disease of both the genome and the proteome combined. And with that, thank you very much.